Okay, this is fine. Hi everyone, my name is Temi, and I'm a PhD student in the IM lab. Today I'll be talking to you about Parsl for parallelized inference and modeling at scale. I added this extra bit to the topic when I was thinking about the story that I want to tell. So let's see. Okay, so importantly, these are very important people. So you might want to take a picture of these names. But I'll be alternating between some science motivation and some computing, and I really hope I don't bore you, but hopefully not. So in genetics, we are mostly concerned a lot, especially in my field, about how we can understand the biology of phenotypes. And so usually we do something called a genome-wide association study, which essentially helps us identify loci or SNPs, in this case, that are associated with a phenotype. So we collect data from hundreds of individuals, we sequence their genomes, we do some quality control, and then we regress these SNPs against the phenotype, and we usually come up with some of these SNPs that are associated with the phenotype statistically, and this helps us generate hypotheses and all of that. Since the first GWAS was published, many more hits or significantly associated SNPs have been published too. So all these tiny dots here are significant SNPs associated with all these phenotypes here, and there's millions of them across the genome. But as biologists, we want to know the functions of these SNPs. And our lab is particularly known to have developed reliable methods to find genes that are associated with a disease. But there are other mechanisms like transcription factor binding, which is what I'm focused on. So for instance, we want to know if a SNP, these CGs or these CCs, if they are if they're associated with a phenotype by affecting transcription factor binding. So does the SNP change how this transcription factor binds, and does that influence the phenotype? And so people have been thinking about this, and this CWAS, which is cis-stroma-wide association study, attempted to find variants that are associated with androgen receptor binding in prostate. And so what they did was they took genotypes of some 130 individuals, and they trained a model to predict whether androgen receptor is going to bind or not bind in prostate tissues, right? So this is what we have in the field. And their method did identify some loci that are associated with androgen receptor binding and prostate cancer risk, right? So this is one SNP where you have this change. So depending on whether the individual has an AA or a GG, it depends. It is associated with androgen receptor peak activity at this locus. So the problem here is that CWAS is not scalable because you need hundreds for you to do this kind of experiment in tissues and for all the transcription factors that we know. We need samples from hundreds of individuals. We need money. We need effort, right? It's consortium level stuff, you know. So I'll jump to this. In a typical machine learning pipeline, we take our data, we train, we test, and we usually, after the model is good, we are able to use the model to do some downstream analysis, right? And so there's this model called Informer that is trained to spit out things that I'm going to be calling the epigenome. Okay, so what you do is you put in the DNA sequence and it spits out like this predicted epigenome. But we know that we can use these epigenetic features to accurately predict transcription factor binding in a cell type. So what I'm showing here is a method that did something similar, and the method is called impact here, and it does perform better than the state of the art at this point. So we intend to use these predicted epigenetic features from Informa to train new models of transcription factor binding, right? So we train here, right? This thing. When I implemented this idea, we ran into this problem where we have this serialization error with Parsl and this TensorFlow model. And I talked to my friend. We just immediately started talking about taking a vacation instead of the issue. So the problem essentially is that Informa, the TensorFlow model, does not serialize properly with Parsl, and also DNA sequence extraction requires many calls, so it's repetitive, right? So we said, okay, we can give each Parsl app its own version of Informa, so which is what we try to do here. So GPU1, GPU2, they will get their own, and then we cache the DNA sequence returned object so that the calls are although repetitive, but it makes use of the same returned sequence extractor. And then right. Thank you. And with the SnakeMake workflow, we scale this up to many transcription factors in many tissues. And to run our workflow, it's really simple. You just SnakeMake file this, and it's fine. And I show here that our method does well. So these are different transcription factors in different tissues, and we predict whether there's binding or there's no binding with these high AUCs. I'm gonna come, yeah, okay. And then we also show that we can. So we trained our method for androgen receptor binding, and we see that we can predict binding or no binding. So each dot here is an individual. And then we compared with CWAS, right, doing this. And we show that our TF-PRED scores match CWAS scores, just noting that we didn't train our method on individuals, we just trained on the reference genome, which is this hypothetical genome. And when we applied our method to prostate cancer risk, we could recapitulate 43 out of the 74 risk loci that they found with our approach. So this is a heartfelt letter to Parsl, saying that we couldn't have done this without you from the IMLab. And in the future, we want to scale this up now to multiple phenotypes in the UK Biobank. And that's what we plan to do to further now let us know a more generalized or comprehensive understanding of transcription factor tissue interactions with a phenotype. And that's it. And thank you. Questions?
Hi, any questions? Then on Zoom? So on the serialization front, Ben Clifford probably has ideas here, but you go by Temi, do you?
Yes, I do.
Yeah, are you on our Slack channel?
Am I? Yes. So I asked questions on the Slack channel, and that was how we were giving the ideas to like cache some of those. Yeah, and that was what actually worked. So in my slide, I showed some memorization stuff like that. But yes, that was what we did. Absolutely.
A lot of these models have functions to actually write them to disk, right? So if you can write the model to disk and reload it on the endpoint, it should work without serialization problems. And then we often see by caching, often it's beneficial in the endpoint, the worker to actually keep that as a global in memory, so you can keep using it based on the inferences that are coming. So yeah, there's a pattern that we're seeing more and more. So we'd love to chat and help you optimize that. So just yell out when you run into things. We can talk about that.
Thank you.